WNT signaling pathway. Introduction. WNTs, wingless related integration site, signaling pathway composed of WNTs, which are secreted factors, plays a very crucial role in multicellular organism development. These secreted factors bind to frizzled receptors which lead to the regulation of various cellular activities like cell division, phase specification, patterning of the axis, and other developmental processes like polarization, segmentation, and so on. Occurrence of a wide range of WNT ligands in its frizzled receptor assists in the generation of signals which is stimulation specific. This pathway is also involved in the auto renewal of somatic stem cells and is also known to be involved in regulating the pluripotency of stem cells. Mechanism and regulation. The responsive target cell possesses frizzled receptors along with a co-receptor LRP56. Binding of WNT ligand triggers activation of DDL, which eventually leads to disassembling of the complex comprises of GSK3 beta axon and AP of the GS3 beta axon, and APC complex prevents beta-catenin degradation, which allows it to translocate to the nucleus to induce transcription of target genes upon binding with the transcription factor TCF/LEF. This is WNT canonical signaling. WNT signaling can be regulated by two beta-catenin independent non-canonical WNT signaling in the absence of WNT. One of those non-canonical pathways is PCP, planar cell polarity pathway, which initiates signaling through frizzled receptors and DDL, primarily involved in cytoskeletal rearrangements that lead to changes in cellular morphology. Whereas the second non-canonical WNT signaling pathway WNT, calcium involved in cellular movement, migration, and adhesion. The signaling initiates upon calcium release through phospholipase. WNT signaling is associated with various cancers as with other signaling pathways involved in cell division and migration, and hence many of the pathway's components are known as tumor suppressors and oncogenes. Physiological significance. The WNT pathway plays a very important role in the development process. In the presence of WNT, frizzled receptors get activated, which eventually stabilizes beta-catenin. Beta-catenin interacts with cadherins at cell-to-cell junction and assists in stabilizing their interaction with the cytoskeleton. Non-canonical beta-catenin has reportedly shown its association with angiogenesis and is involved in the development of blood vessels. Beta-catenin-dependent WNT pathway plays an important role in differentiation and microvascular development in the brain and retina. Because of its involvement in biological processes which sustain cellular growth, differentiation, and integrity, its upregulation in cancer cells is detrimental and hence has been an important anti-cancer target. The cascades in this signaling pathway are closely associated with cancer and retain their stemness and assist in their proliferation. Figure 4.1 illustration of WNT pathway. WNT binding to responsive cells initiates the pathway, which leads to the regulation of various developmental processes by canonical or non-canonical pathways. Hedgehog signaling pathway introduction. Hedgehog signaling pathway gained a lot of attention in the past 2 decades due to its association with developmental abnormalities and the development of cancer due to dysregulation in pathway transduction. The pathway is involved in cellular proliferation and differentiation as well as in developmental processes wherein the pathway decides sulfate and tissue patterning. Hedgehog signaling pathway is also involved in mature tissue homeostasis as well as maintenance of stem cells, tissue repair, and regenerative activities. Mechanism and regulation. The pathway comprises 3 types of secreted proteins: Indian hedgehog (IHH), sonic hedgehog (CHH), and desert hedgehog (patch PTCH), and smooth and smoke regulate activation of the hedgehog pathway. The pathway is often in the absence of hedgehog HH, secreted proteins of smoke, which is a receptor with 7 transmembrane domains, gets inhibited by PTCH. When HH protein is available in the environment, it activates SMO, and a complex made up of Gli and SUFU, which is involved in negative regulation of the signaling pathway, translocates to the nucleus and activates transcription factors Gli1 and Gli2 along with proteasomal degradation of Gli3, a negative regulator of the pathway. Fig 4.19. Activated transcriptional factors eventually lead to the activation of target genes. Figure 4.19 Hedgehog signaling pathway. Diagrammatic illustration of activation of hedgehog pathway in the presence of hedgehog HH secreted molecules, which lead to activation of target genes in the nucleus. Physiological significance. Hedgehog signaling pathway is involved in embryonic development and target genes which regulate cell cycle, apoptosis, and members from WNT signaling pathway. This pathway has long been associated with cancer development, tumorigenesis, as well as other abnormalities like basal cell Nevis syndrome, meningiomas, and many others. The pathway is involved in various stages of cancer development. It could be early stages or could metastatic stages. Hippo pathway introduction. The hippo pathway is a conserved pathway in mammals that was first studied by genetic mosaic screens in Drosophila, and inactivation of the gene resulted in overgrown tissue. This pathway mediated the upstream signals to regulate biological processes downstream and coordinates with different biological processes like cell differentiation, death, growth, and proliferation. Fig 4.20. The upstream elements consist of G protein coupled receptor (GPCR), signaling and cell polarity, which mediates signal to hippo signaling through the actin cytoskeleton. Figure 4.20 Hippo signaling pathway. Diagrammatic illustration of hippo pathway modulating various downstream biological processes like proliferation, differentiation, cell death, and growth. This eventually regulates organ homeostasis, its development, and its size. Mechanism and regulation. Protein kinase MST1/2 phosphorylated LAS1/2, which is increased through phosphorylation of SAVE1 with the help of serodomains present in both LAS1/2 and SAVE, leads to activation of LAB1/2. MOB1 phosphorylation by MST1/2 MOB1 leads to its binding to an autoinhibitory domain in LAB1/2 and increases its kinase activity. SAVE1 interaction with LAB1/2 could increase or suppress its activity as cell type dependent. Further downstream, LAS1/2 phosphorylates YAP-TAZ having the consensus sequence HXRXXS. While phosphorylation of YAP-TAZ leads to its protein degradation in the cytoplasm by interacting with the 3rd of March 14, inactivation of its upstream kinase keeps YAP-ZAP in its hyperphosphorylated state, resulting in its translocation into the nucleus and playing a role in its gene expression. Fig 4.21. They don't bind to DNA directly. Instead, they bind to transcription factor complexes and assist in regulating genes involved in cell proliferation, growth, differentiation, and death. Figure 4.21 Hippo signaling pathway. Diagrammatic illustration of hippo pathway components. MST1/2 phosphorylates SAVE and MOB, which assists in LAS1/2 phosphorylation with the help of protein kinase MST1/2. Phosphorylated LAS1/2 phosphorylates YAP-TAZ leading to its sequestration in the cytoplasm by interacting with the 3rd of March 14. Under the non-phosphorylated and hypophosphorylated conditions, YAP-ZAP translocates to the nucleus and assists in gene expression by binding to transcription factors. Physiological significance. Adhesins are the proteins that are involved in cell-to-cell adhesion. Some of these are known components in the hippo pathway. Many of the regulators of the hippo pathway are known to regulate apical basal polarity in epithelial cells. Some regulatory components of epithelial cells perpendicular to the apical basal axis (PCP) are known to interact with the hippo pathway and regulate it in Drosophila. 2 networks, namely frizzled flamingo and fat Daxaus, are involved in generating PCP. Fat Daxaus is shown to regulate the hippo pathway. There are reports suggesting regulation of the oncogene YAP-ZAP by growth factors, hormone factors. Hence, YAP-ZAP can be regulated by extracellular or diffusible cell surface receptors which are involved in interacting with growth factors. G protein coupled receptor (GPCR) is one of the known regulators of YAP-ZAP and controls cell proliferation and differentiation. YAP-ZAP is involved in various important cell biological functions like proliferation and migration, and hence its upregulation in breast cancer has shown to be involved in enhancing tumorigenesis. YAP-ZAP is shown to be sensitive to mechanical cues obtained from cell spreading during migration, proliferation as well as in cancer cell metastasis, extracellular matrix stiffness during cell migration, and tumorigenesis, and hence has been an important therapeutic target in anti-cancer development. Cellular organization and communications. Most of the movements of the cells are coordinated by the actin filaments in association with myosin. Here myosin is a protein that behaves like a molecular motor that converts the chemical energy to mechanical energy. This conversion is very important for producing force and movement, whose application is found in muscle contraction. Muscle contraction gives us an appropriate model for learning the concepts of interactions between actin and myosin as well as the myosin molecules which exhibits motor activity. There is a number of applications of the interaction of actin and myosin other than muscle contraction. They include all the non-muscle cell movements like cell division and crawling movements of cells on the surface. Major movement is due to actin polymerization. Now when we talk about the cytoskeleton, actin is a major protein that contributes to the cytoskeleton of the body. Actin filaments of about 7 nanometers diameter are formed by the polymerization of these actin proteins, which are, in turn, again get arranged to form more compact structures like bundles or 3-dimensional crosslinks. The various dynamics of the actin-like polymerization, as well as the disintegration process is under the tight control of actin binding proteins. So we can say that the actin-based cytoskeleton, made up of actin filaments and actin binding proteins along with a number of myosin motor proteins, is mainly concerned with the various kinds of motility. The actin filaments are present below the plasma membrane in a profuse amount. This region is of high actin filament density and is commonly found at the cell periphery, which is known as the cell cortex. They form an arrangement that is instrumental in providing mechanical support to the cells, determines the shape of the cell, and permits the cells to move so they can divide the food particles and also migrate to different places as per the requirement. The actin is expressed in various isoforms in the higher eukaryotes like alpha, alpha, beta, beta, and gamma, gamma, depending upon their isoelectric point. Normally, alpha actins are found in muscle, while beta and gamma isoforms are the major constituents in the non-muscle cells. The observations and the results depict that when there is formation of single actin monomers, the 2 actin monomers align themselves in the asymmetric orientation to form a two-fold axis of symmetry and finally congregate into a filament that is made up of a pair of strands causing a left-handed helical twist when the adjacent subunits are positioned with respect to each other. The diverse cellular processes like endocytosis, cell migration, and division of cytoplasm all require the dynamic reorganization of the actin filaments. These processes actually provide a number of actin structures that endow the cell with the property of plasticity, which may alter the cell structure according to the signals obtained. The external stimulus actually regulates the temporary polymerization of the actin fibers, resulting in cell motility. The network of the actin filament formed actually forms the basis of the rigid structure, with the individual filaments branching off the sides of others. Assembly and disassembly of actin filaments. When the actin filaments organize into a compact network, a structure called the lamellipodia extends, forming a protrusion from the living cell. The protrusive movement of the cell is maintained by the quick assembly and disassembly mechanism. The actin filaments or the microfilaments are reorganized into different structures, resulting in the formation of bundles or three-dimensional networks which exhibit the properties of stem cell gels. There are a number of actin binding proteins which control the assembly and disassembly of actin filaments. Further cross-linking these to form bundles and networks as well as their alliance with the other cellular structures like plasma membranes. The association of the actin filaments with the plasma membrane is very essential for providing about 95% of the amino acid sequence of actin protein is conserved. The actin monomer, the G-actin, or globular actin polymerizes with other G-actin molecules at its tight binding sites, resulting in a head-to-tail connection forming the filamentous actin or F-actin. An angle of 166 O is formed between each of the G-actin, leading to a twisted helical manifestation in the filamentous or F-actin. A distinct polarity is provided to the filaments of actin due to the same orientation of the actin molecules in the polymer with one end, which grows to the plus and the minus end to the terminal part from where the G-actin gets dissociated. The polarity of actin filaments is of great significance in the assembly of actin fibers and also provides a particular route for the movement of myosin. Shape to the cell as well as mechanical support and movement. In the year 1942, actin was isolated from muscle cells for the first time, and since then, it has been considered a major protein in muscle cells only, but presently it has been established that it is one of the most common proteins present in most eukaryotic cells. Only one gene encoding for actin protein which is conserved is present in yeasts, while the eukaryotes, like mammals, have 6 different types of genes, of which 4 are expressed in muscle cells and the rest 2 in non-muscle cells. Kenneth Holmes et al. In 1990 determined the three-dimensional structures of actin molecules and actin filaments. Actin is a very rich protein formed of 375 amino acids with a molecular weight of about 43 kDa. Figure 4.22 Assembly and disassembly of the actin filaments. The actin monomer, the G-actin or globular actin, polymerizes with other G-actin molecules at its tight binding sites, resulting in a head-to-tail connection forming the filamentous actin or F-actin. The dynamic nature of the actin filaments and their polymerization is in direct correlation with their disassembly. The polymerization of actin filament takes place in 3 phases. The first phase being the nucleation phase, then comes the elongation phase, and lastly, the steady state phase. So the polymerization of actin protein initiates with the formation of an actin nucleus, which is formed by the aggregation of 3 actin monomers to form a small complex. The next phase, the elongation phase, starts with the addition of monomers to the filament at the barred end or the plus end in the presence of elongation factors like formin. What is actin nucleation? The nucleation is the first step in actin polymerization. In this step, the actin nucleus is formed, which consists of about 3 of the actin monomers. This forms the basis for the elongation of the actin filament. The presence of G-actin monomers alone are not able to form the actin nucleus because of the instability of actin oligomers. So, that means some kind of supplementary factors are required for the nucleation process. Models for actin polymerization. 2 models have been proposed to explain the mechanism involved in the polymerization of actin filament in the presence of supplementary factors. The first model, which is known to be the tip nucleation model, involves the foreign proteins complex present in the membrane, which mediates the filament extension stabilized by cross-linking by Cas proteins. Figure 4.2 creates a schematic of the tip nucleation model of actin nucleation. This involves the foreign proteins complex present in the membrane, which mediates the actin filament extension stabilized by cross-linking by Cas proteins. The other model that has been suggested is known as the convergent elongation model, which says that the arc 2/3 complex is found to be essential for the initiation or nucleation of phylopodia. The nucleated branches of the arc 2/3 complex persistently build up from the network of the actin filament. These networks are located at the foremost periphery of the lamellipodia, which gradually converges to form a complex protected by the cross-linking and direct nucleation model tells us that a nucleation factor, e.g., MDIA1 aggregates the new filaments at a particular site near the protruding edge, such as the focal adhesion. At the time of filament elongation, bundling formin displaces the barred end-bound nucleation factor, e.g., FMNL3 or MDIA2, resulting in further elongation, as well as bundling, to drive productive phylopodial growth. According to still another new concept, the extended convergent elongation model, the actin filaments are aggregated at a particular site by a nucleation factor such as MDIA1 and serve as mother filaments for arc 2/3 complex mediated nucleation. Bundling formins, such as FMNL3 and MDIA2 then mediate convergent elongation on the arc 2/3 complex nucleated barred ends of the FASN protein. The arc 2/3 complex alone is a very incompetent nucleator as its activation requires binding of the actin filament to the nucleation promoting factors, NPFs, that have WCA domains consisting of G-actin binding WH2W domains and arc 2/3 binding central acidic CA. Once the arc 2/3 complex is activated, the nucleation is initiated and the formation of new filaments is started from the sides of the original filaments at an angle of 70 degrees so as to form a Y-shaped network. On the other side, it has been established that ATP hydrolysis by actin in the arc 2 subunit of the arc 2/3 complex and binding of core protein supports to the branching of the actin filament and recycling of the arc 2/3 complex. Figure 4.24 a schematic depicting the convergent elongation model of actin nucleation. From the actin filament network located at the leading edge of the lamellipodia, these filaments are proposed to gradually converge, forming a bundle that is secured by Cas cross-linking. The arc 2/3 complex, which is more commonly associated with lamellipodia formation but has been found to be critical for phylopodia initiation, plays a role. Here, arc 2/3 complex nucleated branches continually develop. Figure 4.25 models for phylopodial assembly. Model A convergent elongation and B tip nucleation, C indirect tip nucleation model. A nucleation factor, e.g., MDIA1, assembles new filaments at a specific site near the leading edge, purple oval, such as the focal adhesion. During filament elongation, the barred end-bound nucleation factor is displaced by a bundling formin, e.g., FMNL3 or MDIA2, which allows further elongation, as well as bundling, to drive productive phylopodial growth. D extended convergent elongation model. Actin filaments are assembled at a specific site, purple oval, by a nucleation factor such as MDIA1 and serve as mother filaments for arc 2/3 complex mediated nucleation. Bundling formins such as FMNL3 and MDIA2 then mediate convergent elongation on the arc 2/3 complex nucleated barred ends. We do not depict VASP in these models because its role is still unclear, but it could, in principle, enhance the initial assembly of filaments in models A, C, and D. Elongation of actin. The polymerization of unbranched actin filaments is catalyzed by a very important group of proteins called formins. The interactions between actin and the FH2 domains of formins like CDC12, BNI1, and MDIA1 have been studied to establish the mechanism of elongation. There are 2 factors that regulate the elongation process of the actin filaments. The first being the FH2 domains can sterically obstruct the adding up of new actin subunits, and the second involves the flattening of the FH2 domains preventing role in both nucleation as well as elongation of the actin filament as well. The progressive relationship of formins with the growing ends permits the addition of actin subunits and also checks the termination of polymerization by the capping proteins. The subunit addition at the pointed end of the actin. Formin proteins are the second type of chief complex of actin nucleators. Unlike the arc 2/3 complex, the formin proteins function as multi-domain proteins that, as dimers, work to assemble the unbranched actin filaments. The formin protein complex plays a significant. Figure 4.26 elongation of actin filaments. NPFs, e.g. WASP, SCAR bring together the arc 2/3 complex and actin monomers to nucleate new actin filaments and form new branches from the side of pre-existing filaments. Arc 2/3 complex remains at the minus end of the filament. B formin cooperates with profilin to nucleate new actin filaments. Formin remains at the plus end of the filament. The regulation of elongation of actin filament at the barred end is very critical for the generation of force by actin networks. 3 different mechanisms involving arc 2/3 derived networks have been established that control the elongation of the actin filament at the barred end. AIP1 protein prevents the ADP actin filaments elongation complex with covalent protein along with capping protein caps maintaining an elevated concentration of actin which is said to be assembly competent. ABP1 and AIM3 are 2 additional proteins that work in association to prevent the elongation of the barred end. ABP1 AIM3 associates with capping protein CAPC so as to regulate the elongation of newly organized ATP actin filaments to bestow the property of the filament polarity in the network of actin. So we can witness 3 different mechanisms which regulate the elongation of the actin filament in different areas of arc 2/3 networks. CAPC caps some F actin at the plus end. Profilin binding activates actin binding to the plus end. Profilin plus dash arc complex ATP G-actin. Thymosin binding inhibits actin association. Arc 2 arc 3 complex nucleates actin polymerization. After polymerization, actin hydrolyzes its ATP but keeps ADP tightly bound. G-actin is activated by binding ATP. Thymosin. Capping protein CAPZ. Tropomodulin caps some F actin at the minus end. Plus. Figure 4.27 polymerization of actin. When thymosin is present, the actins are sequestered by them, and hence it inhibits the association of actin, whereas profiling, when bound with actin, enhances the polymerization of actin. Tropomodulin protein caps F actin at the minus end of the growing actin filament, and cap C actin at the plus end. Actin nucleators are required by the cell so as to catalyze the assemblage of filaments de novo and also factors for actin elongation to regulate the speed and degree of polymerization. The above factors are enumerated as ART 2/3 complex, Forman's, ENF BASF, and newcomer Spire, Cobalt, and LMOD. Actin filament assembly. Polymerization. Trimer. Formation. Dimer. Formation. Nucleation at high covalent concentrations. pi release. From filaments, debranching. Severing. At low covalent concentrations. Stable filaments. When there is no covalent, the actin filaments are stable. When the concentration of covalent is less, severing of the actin filament takes place, leading to disassembly of the actin filament. But when the concentration of covalent is high, then nucleation of the actin filament takes place, leading to the assembly of the actin filament. Steady state of actin polymerization. The steady state phase witnesses the entry of the filament in a state of equilibrium where a balance is maintained in the disassembly of the monomer from the end of polymerization at the plus ends. This balance is maintained by critical cytosolic concentration of monomers of actin. This steady state assembly and disassembly of the actin filaments is known as treadmilling. As we know that the actin network is made up of filamentous actin or F-actin. The aggregation or assembly is fueled by ATP hydrolysis with the occurrence of filament nucleation instinctively in vitro. The polymerization or the addition of ATP actin occurs at the barred end, which results in the elongation of the filament. The process of elongation will persist till the rate of elongation is larger than the loss of ATP actin from the pointed end. The profiling protein binds to the ATP actin, thereby preventing the nucleation process and acceleration of the elongation of the filament in vivo. Now depolymerization occurs when the ATP actin dissociates at a faster rate than the rate of ATP actin association, thus resulting in a shortening of the actin filament. Covalent protein helps the shortening of actin filament in vivo by shearing off the filaments into short fragments, promoting the loss of subunit from the pointed ends. So now the process of actin treadmilling takes place, marking the balance in the rate of association of ATP actin and the rate of loss of ATP actin. Figure 4.29 Nucleation, elongation, and steady state phase of actin filament assembly. During the nucleation phase, the formation of a stable actin nucleus occurs. This is usually comprised of free actin monomers in the complex. In the elongation phase, monomers are rapidly added to the filament at the plus A or barred end, and this is often facilitated by additional elongation factors such as formin. For this process to occur, the plus end of the filament must be exposed, and this means the removal of capping protein. The depolymerization of the actin filament. The depolymerization of the actin filament maintains a pool of monomers of actin and allows the continuous reorganization and growth of the cytoskeleton of actin. Disassembly of actin filaments takes place at the vein end or at the pointed end of the filament in the presence of ADF covalent proteins. Essentially, the dissociation of the actin monomers from the barred end takes place at a faster rate than from the pointed end. The covalent protein present at the pointed end of the filament is instrumental in destabilizing the filament, releasing the actin monomers complexed with ADP. This action of the profiling protein is counteracted by the presence of the formins capping proteins at the barred end, thus resulting in a more stable filament. The ATPF actin to ADPF actin conversion requires the hydrolysis of ATP, thereby releasing free inorganic phosphate pi molecules that bind to the covalent protein, which in turn binds to F actin. The complex of covalent with F actin and pi, when it binds to the actin filament, then gets destabilized by conformational change which produces a twist within the filament. This exerts a kind of strain resulting in the injury to the integrity of the fiber and an increase in the rate of pi by about tenfold, thus leading to disassembly. It was also established that the efficiency of covalent in the shearing of the actin filament is increased in actin bundles even though the binding of the covalent to the facet bundled actin filaments is decreased. This improved efficiency permits the shearing of bundled actin filaments at concentrations of covalent that are inadequate to induce the severing of free filaments. The reason for this increased efficiency has been related to the facet of the protein crosslinkers. It has been anticipated that the facets reduce the flexibility of trunculanes obtained from the Red Sea sponges bind to the actin monomer and inhibit polymerization, thus promoting filament disassembly. Jasglycan light from marine sources stabilize the actin monomer, thereby enhances filament nucleation and assembly. A drug called colloidin affects the actin filaments by attaching tightly to them and inhibiting their dissociation into individual actin molecules. Organization of actin filaments. The assembly of the actin filaments can be classified into two categories. The first one is a structure called the actin bundles, and the other one is the actin network. The actin bundles contain actin filaments cross-linked to each other closely into parallel arrays. These actin bundles support various projections of the plasma membrane, like the microvilli. All the filaments in these bundles have the same polarity, with their plus ends adjoining the plasma membrane. Fibrin, a 68 kDa actin bundling protein that is concerned with the formation of the bundles, was for the first time isolated from microvilli of the intestine, and then they were afterwards located in the surface projections of different cells. This protein consists of two adjoining activating domains, which cause the binding of the actin filaments in a bundle. The actin bundling proteins that aid in cross-linking actin filaments into bundles are small rigid proteins that compel the filaments to line up closely with one another, while the proteins that arrange the actin filaments into networks are large flexible proteins that can cross-link perpendicular filaments. These actin cross-linking proteins are complex proteins formed from similar structural constituents. The activating domains of these proteins are very light in structure and are separated by spacer sequences that have variable length and flexibility, and these are actually responsible for the unique cross-linking properties of different actin binding proteins of the actin filaments and thus making them more susceptible to the twisting effect of covalent. Environmental factors such as pH also play a crucial role in the depolymerization of the actin filaments, whereas higher pH favors the depolymerization of the actin filaments. The reason for the depolymerization is the weaker binding of pi in an environment which is more alkaline. The Acs have more affinity for ATPF actin than ATPF actin, which causes the shearing of the actin filaments in the innermost regions of filaments as that region is rich in ATP actin. Drugs that influence the actin polymerization. A number of drugs from different sources have been found to play a major role in maintaining the dynamics of the actin filament. These drugs are very significant in the study of the assembly and disassembly kinetics of actin. They bind to the actin at different sites and influence the polymerization process of the actin filament. There is evidence that latrunculin disrupts the polymerization of pure actin in vitro in a manner consistent with the formation of a 1 molar complex between latrunculin A and G actin. Actin targeting drugs such as cytochaslin D and jasglycan light have shown great promise as antiproliferative agents in vitro. Latrunculin A LATA is a toxin obtained from the Red Sea sponge Latrunculia magnifica and is used in the depolymerization of actin filaments. LATA binds actin monomers and sequesters them from polymerization. Low concentrations of LATA result in the fast disassembly of actin filaments in animal and yeast cells. Depolymerization is usually assumed to result from the sequestration of actin monomers. The cytochaslins obtained from the molds bind to the barred end of the actin filament to prevent actin filament assembly and disassembly. So this drug induces changes in cell shape as well as inhibition of cellular movements like cell division following mitosis, which reveals that actin polymerization is required for these processes. Actin. Filament. Filament fibrin. Actin. Proteins. Figure 4.30 Polymerization and depolymerization of actin filaments are under the control of many actin-associated proteins. In this image, several organizations of actin filaments are depicted, as well as the actin-associated proteins that influence these arrangements. The second assembly, the actin network, consists of the actin filaments loosely cross-linked or conjoined in orthogonal arrays resulting in the formation of a three-dimensional network possessing semi-solid properties. There are certain actin binding proteins which aid in the cross-linking of the actin filaments into different patterns. These proteins possess two distinct domains, one of which binds the actin and the other domain helps them to cross-link with other actins forming a network. Just like fibrin acts as an actin binding protein in the actin bundles, alpha-actinin, a 102 kDa protein, binds to actin as a dimer in the actin networks. Each subunit contains a single actin binding site in contrast to fibrin, which has two binding sites. The filaments which are cross-linked by alpha-actinin are a far distance of 40 meters apart than the ones which are connected by the fibrins, which are 4 nanometers apart. The rationale behind the greater spacing between the filaments in the actin network is that it aids the motor protein myosin in interacting with the actin filaments in these bundles. Filament, a 280 kDa actin binding protein or ABP280, is responsible for cross-linking of the actin filaments in a network. The filament dimer is a flexible V-shaped molecule with actin binding domains and dimerization domains, or at opposite ends of each subunit. Consequently, the filament forms a cross-link between orthogonal actin filaments, creating a loose three-dimensional meshwork. Myosin is enzyme. Myosin is the main constituent of the thick filaments, and each of the thick filaments is constituted by about 250 molecules of myosin. Myosin has two significant functions, the first being the structural function, where it acts as a building block for the thick filaments, and a practical one, where it acts as a catalyst in the breakdown of ATP during contraction and in its interaction with actin and the role of force generator of muscle. When we talk about the structural organization of one particular myosin molecule, we witness that it consists of two main protein chains and four smaller chains. The myosin molecule as a whole is about 160 nanometers in length, having a symmetrical arrangement with a 120 meters long rod-like tail consisting of two protein chains wound into an alpha helix, forming a coiled structure. Two globular head-like regions made up of two protein chains are present at the extreme end of the molecule and have the ability to attach to the actin protein. This particular head region carries the enzymatic sites for ATP hydrolysis. The myosin head attaches to the binding site on the actin filament. In addition, it binds ATP, acting as an enzyme to transfer energy from ATP. The energy transfer changes the shape of the myosin head, COX, the apparatus. Myosin is an ATPase that travels along actin filaments by coupling the process of the hydrolysis of ATP to conformational changes. So myosin is a type of enzyme which actually converts chemical energy into mechanical energy and thus is called a mechanochemical enzyme or a motor protein. Pollard and Korn, in 1973, discovered that there are some enzymes in acanthamine castellane with myosin-like functions. After this discovery, a number of myosin genes have been established that express different myosin proteins with ATPase activity. Previously, the function of myosin was considered to be restricted to muscle cells, but later it was discovered that various myosins are the protein products of a very large superfamily of genes possessing the properties of actin binding, ATP hydrolysis, ATPase enzyme activity, as well as force transduction. Figure 4.31 Myosin ATPase cycle. Most of the dozens of myosin isoforms, including skeletal, cardiac, smooth muscle, and non-muscle, follow the force-generating cycle diagram below. First, steps A and B, myosin motor domain has hydrolyzed ATP to ADP and inorganic phosphate pi. At this stage, myosin heads bind weakly to actin filaments. Next, step C, myosin releases phosphate. At around the same time, myosin's lever arm swings to produce the force-generating power stroke, turned working stroke in this preprint. Finally, step D, myosin heads exchange ADP for ATP and release actin to restart the cycle. Also, Perry, and Thomas in 1974 concluded that the myosin light chain acts as a kinase and differs in several properties from phosphorylase kinase and protein kinase and represents an enzyme not previously reported in the literature. The systematic determination by Michael Bahrain of the relationship between the speed of muscle contraction and myosin ATPase activity had been established. The investigators, being influenced by the Bahraini's finding that myosin ATPase activity is an essential driver of contraction speed, have developed small molecule modifiers of myosin turnover kinetics for the correction of the altered contractility of cardiac muscles due to inherited or acquired myopathies. According to Malcolm Murray in 2017, the discovery also finds new avenues in the field of medical therapeutics where an increasing number of molecular targets for potential therapeutic interventions involving myosin is based on the discovery of inherent modulators of the accessibility of myosin for communication with actin in skeletal and possibly cardiac muscle. Cell crawling. A variety of cells exhibit movements like crawling, which is the most basic form of cell locomotion on the surface of the substratum. The movements are shown by amoebas, movement of the white blood cells towards the tissues in order to find an infection. Metastasis of malignant tumors, etc. are a few examples of the movement of cells which involve crawling. Phagocytosis and extension of nerve cell processes during the nervous system development also involve the movements of cell crawling. A cell's movement is motivated by persistent reconstitution of the cytoskeleton and is mediated by the tiny filaments composed of a protein called actin called lamella located at the front edge of the cell, known as lamella. A pushing force is achieved when actin subunits are added to the lamella by the process of polymerization as opposed to the addition of the molecular motor, myosin 2, which results in the occurrence of a contractile force. The result of pushes and contraction actin filament cytoskeleton results in the crawling of the cell. The movement of cells along substrates outside the cell surrounding and substratum needs an extremely small force for the cell to move against the friction between the surface of the cell and the surrounding liquid. The force required for the movement must be produced from the cell themselves, and no other agency can generate the forces. Cells crawl due to the arrangement of actin filaments within the cell cytoplasm. Various cellular events like inflammation, clotting of blood, healing of wounds, and the metastasis of cancer all involve the phenomenon of crawling. These cells are released into the circulatory system after being produced in the bone marrow and then, in due course, crawl out of capillaries and into the tissues to demolish probable pathogens. Lipincourt Schwartz et al. In 2011 demonstrated the crawling of a live cell with the help of videos in which she described the dynamics of the actin reorganization and related them to the fundamental mechanics of cell motility. They proved that the base against which the lamella is polymerizing actin filaments protrude is formed by the actin arcs of the lamella. The machinery for the movement of cells is actually the combination of two self-assembling molecular systems, which are continuously undergoing cell movement and are designed as two self-assembling molecular systems undergoing persistent turnover of the actin cytoskeleton inside the cell. The actin cytoskeleton is the force-producing machinery and consists of filaments made of protein actin and numerous accessory which are transmembrane structures present in the plasma membrane. This system actually acts as a mechanical link mediator between the cytoskeleton and the extracellular substrate. The movement of the cell takes place in two steps. The first step is the protrusion of the cell's leading edge, which is produced by the self-assembly of the actin filament network at the front of the cell and the adhesion molecules beneath. The second step is the withdrawal of the cell back, demanding the dismantling as well as the removal of the two systems from the back part of the cell. The mechanisms of self-assembly of the actin cytoskeleton at the cell front and generation of the forces have been extensively investigated, while the dismantling and reprocessing of the two systems during the retraction process are still being investigated. Figure 4.32 Crawling locomotion of keratocytes fragment. Crawling proceeds through the protrusion of the cell's leading edge, is driven by the polymerization of the actin network interacting with the substrate through cell adhesion. The actin network density decays toward the cell rear, where its remnants get crashed through force application by the cell membrane. This force is produced by the actin assembly of the cell's leading edge and transmitted to the cell's rear through the membrane. The cell movement is enabled by detachment and recycling of the adhesion at the rear edge. The movement of cells is governed by the gel salt theory put forward by Hyman, 1917, and supported by Panting and Mast, 1926. According to this theory, the cells exist in a gelsal state, which means that at any moment, some of the regions of the cell are stiff gel and other regions more fluid natured sol. The fact is that the interior region of the cell is usually in a fluid state or sol state and a gel or rigid state at the periphery. In order to acquire a crawling movement, a weak area is created in the periphery region of the gel region, and then the sol region is forced into the weakened area, which results in the formation of a pseudopod or false speed-like structure. The pseudopod now maintains the gel state with the assembly of the actin filaments. With the help of certain membrane proteins, the pseudopods stick to the surface and the cell crawls on, pulling the rest of the cell in the direction of the pseudopod. This phenomenon of developing a pseudopod needs some kind of force which is created when actin filaments in the end opposite to the cell contract. This condition resembles the event of squeezing a water balloon at one end, giving rise to a bulge at the other end of the balloon. Steps in the crawling movement. Cell crawling is a synchronized cycle of movements in three stages. The first stage involves the formation of projections like the pseudopodia or lamellipodia that is actually the part from the leading edge of the cell. So motility in a cell is originated from an actin-dependent protrusion of the cell's leading edge. This protrusion contains actin filaments, with elongating barbed ends aligned towards the plasma membrane. In the second stage, the extensions must adhere to the substratum of the cell on which it is migrating. This attachment is mediated by the proteins present in the membrane of the actin. The final step involves the dissociation of the trailing edge from the substratum and its retraction into the cell body. A kind of tension is developed between the front and rear end of the cell, creating some kind of contractile which finally drags the rear end of the cell forward. The mechanism involved in cell crawling and crawling movements are based on the dynamic properties of the actin cytoskeleton. The polymerization and cross-linking of actin filaments result in the formation of the protruding edge. This fact has been demonstrated by treating the actin with cytochastolin, which is an alkaloid that prevents actin polymerization, hence blocking the formation of projections on the cell surface. The projections like filopodia and lamellipodia at the protruding edge of the cell appear due to the controlled coordination of the dynamics of actin filaments. The proteins like cofilin and ARC2/3 proteins are involved in the execution of this process. Myosins like myosin are found to play an active role in the pseudopod extension in the amoeba, and myosin B is required for the formation of neuronal filopodia. Capping protein. Actin polymerizing facet module. ATP actin ADP actin. Foramen. Initiation protrusion bundling. Filament. Bundling elongation. Fast. Elongation actin. Assembly profiling. Retraction. Retraction. Retrograde flow. Figure 4.33 Filopodia formation. Actin filament assembly can be initiated by uncapping preformed actin filaments or by de novo formation, which includes both foramen and ARC2/3 mediated, not shown, nucleation. The force produced by actin assembly at the barbed end of actin filaments drives membrane protrusion. The growing end of an actin filament is oriented towards the cell's leading edge and composed of high concentrations of ATP actin. Numerous proteins, including IRSP53, ENF-BASP proteins, and WASP-SCAR proteins cooperate to promote actin assembly and enhance the bundling of actin filaments by FASN. When the barbed end of the filament is capped, this stops filament assembly and protrusion. Continued retrograde movement of the filaments results in retraction of the filopodium. Cytoskeleton. The cells must sort out themselves in the space and interface with their condition to work appropriately. They should be effectively molded, strong physically, and appropriately organized. Numerous cells alter to form and migrate to different capabilities from one location to another. All cells need to rework their interior segments as they develop, partition, and adjust to changing intra and additional cell conditions. Cells in eukaryotes have built up these entire spatial and mechanical capacities to an exceptionally high degree, and they rely upon a striking arrangement of filaments called the cytoskeleton. The cytoskeleton is basically an intricate system of interconnected filaments and tubules that reaches out all through the cytoplasm, from the center to the membrane's inner surface. Cytoskeleton implies important jobs in cell division, growth, and transports membrane-limited organelles within the cytoplasm efficiently in eukaryotes. The cytoskeleton is likewise engaged with numerous types of cell movement and is recognized with various methods, for instance, cell marking and cell attachments with another cell. Different events on the cell surface change the cytoskeleton, and at the same time it seems to take an interest in and modify these activities. The term cytoskeleton actually communicates the role of this polymer network arrangement in giving compositional structure for various functions performed by the cell. It helps in maintaining the internal cell organization and empowers them to expect and keep up their complex shapes. The three noteworthy structures, microtubules, microfilaments, and intermediate filaments, are components of the cytoskeleton in eukaryotes. Microfilaments are approximately 7 nanometers in diameter and are actin protein polymers. In the spectrum of 8 to 12 nm, intermediate filaments have diameters, and their subunits differ by sort of tissue. Electron microscopy has researched the three kinds of filaments and tubules. Further biochemical and cytochemical studies recognize the different explicit proteins of every framework. Particular proteins inside the cytoskeleton were localized with the help of indirect immunostaining. Each fundamental component of the cytoskeleton has a trademark volume, composition, and dispersion, and each element is represented by the polymerization of an alternative subunit kept by non-covalent bonds together. Cytoskeletal filaments also have many other associated proteins. The cytoskeleton is sorted out into distinct structures, principally gel-like cross-sections or in the form of bundles, etc. Actin microfilaments and intermediate filaments are generally joined to membranous proteins and make a skeleton that aids in bolstering the plasma membrane. Also, actins participate in several types of cell motions, although intermediate filaments are not legitimately engaged in cell motion. Microtubules (MTs) are profoundly unique cytoskeletal polymers made from alpha-beta tubulin dimers, and regulation of intracellular MT dynamics is important for several biological processes, such as the attachment and segregation of chromosomes during mitosis and MT stabilization of migrating cells. Several findings have shown that bacteria, including rod-shaped bacteria and the archaea, have polymer systems that work very close to microfilaments, microtubules, and intermediate filaments. It is clear that they play roles close to their eukaryotic counterparts based on the variety and multifaceted nature of MT arranged in various cell types has been uncovered by fluorescence microscopy. Cytoplasmic MTs perform various functions. They maintain the axons in animal cells. In animals, some moving cells require cytoplasmic MTs to keep up their polarized shape. However, in plant cells, cytoplasmic MTs oversee the direction of cellulose microfibrils saved during the development of cell walls. Cytoplasmic MTs structure the spindles during mitosis and meiosis, which is actually essential in the process of mitosis via chromosomal movement. Cytoplasmic MTs organize filaments to control vesicles and various organelles movement. The second set of MTs, axonal microtubules, incorporate the extremely arranged, robust microtubules observed in cell development related specific subcellular constructions, including cilia, flagella, and the basal bodies to which these extremities are attached. The focal shaft, or axonium of ciliary flagella, consists of a heap of exceedingly requested axonal MTs and associated proteins. The sperm shaft axonium also includes MTs. Microtubules consist of protein tubulin and are at a range of approximately 25 nanometers. Results of mutating these proteins. For instance, the actin-like mREB protein is implicated in DNA segregation. The tubulin-like FTSC protein is engaged in deciding where bacterial cells are going to divide, and the intermediate filament-like crescentin protein is an essential controller of cell type. Considerably, certain eukaryotes, such as chloroplasts and mitochondria, generate the FTSC protein by certain organelles and located at sites where these organelles split. While the bacterial proteins at the amino acid level are not very similar to their eukaryotic counterparts, X-ray crystallography has shown that their overall structure is remarkably similar when assembled into polymers. The related proteins also bind the same phosphonucleotides as their eukaryotic counterparts, suggesting striking biochemical similarities. Structure of microtubule. MTs are the largest cytoskeletal elements, table 4.2. In animal cells, MTs are classified into two subgroups, differing in organizational level and structural stability. The main group contains a loosely sorted out, powerful system of cytoplasmic microtubules. Also, the decent microtubules, microfilaments, intermediate filaments. Structure empty tube and wall consisting of protofilaments 13 and no. 2F actin interlocked chains. 8 protofilaments ended with lurch overlapping. Monomer units alpha tubulin, beta tubulin, G actin many proteins. Function chromosomal movement. Organelle movement. Upkeep of animal cell. Structure, motility of cells. Cell motion and division of cells, muscle contraction, cytoplasmic streaming, retaining the form of animal cells. Retaining the form of animal cells. Structural support. Strengthening of axons, formation of lamina of the nucleus. Diameter inner 15 nanometers, 7 nanometers, 8 to 12 nm. Outer 25 nanometers, table 4.2 basic structure and function of microtubules, microfilaments and intermediate filaments. Tubulin subunits. MTs are erect, empty cylinders with an inner distance of approximately 15 nanometers and an outer diameter of approximately 25 nanometers. The length of MTs changes largely. Some are under 200 nanometers long. Others, for example, axonal MTs, can be numerous micrometers long. The MTs wall consists of straight polymer longitudinal modules called protofilaments. Mostly, a protein tubulin heterodimers is the fundamental subunit of a protofilament. The 13 protofilaments are arranged side by side around the hollow center or cavity. However, some MTs in a few animals are smaller or higher than 13 protofilaments. The heterodimers, which are the main component of protofilaments, consist of one molecule alpha tubulin and beta tubulin each, Fig 4.34. After individual alpha and beta tubulin molecules are produced, they tie each other in a non-covalent manner to create a tube heterodimers that does not separate under normal conditions. Both approximately 4 to 5 nm diameter and 55 kDa molecular weights have a single diameter for the tubular molecules. Basic investigations demonstrate that alpha and beta tubulins have almost indistinguishable three-dimensional structures, despite the fact that the similarity to the amino acid sequence is only 40%. Another place in the middle to which colchicine combines colchicine, MT poison which hinders MT placing, and free space in the C terminal that interacts with entylated protein snaps are located on each tubulin subunit. All of the tubulin dimers within the microtubule are arranged in a similar way, with the aim that all of the alpha tubulin types face the same extreme. This standardized direction implies that the protofilament contrasts chemically and constructively with one edge of the protofilament and that the protofilament has a feature of intrinsic polarity, which is therefore comparable to the protofilament itself due to the location of the tubulin dimer in an MT. A lot of living beings have different yet related genes for the subunits of alpha and beta tubulin. Such as, there are 5 alpha residue and 5 beta tubulin isoforms in the brain of a mammal. These isoforms mainly contrast the C-terminal domain, which recommends that several isoforms of tubulin cooperate with different proteins. Tubulin can be adjusted synthetically regardless of extraordinary isoforms. In particular, the tubulin acetylated is structurally more stable than the tubulin non-acetylated. Figure 4.34 Structure of microtubules. MTs are vacant, upright, exterior diameter cylinders of approximately 25 nanometers and inner diameter of approximately 15 nanometers. Figure 4.35 Structure of microtubule monomers. Diagrammatic representation of the numeric tubulin subunit in the form of ribbons. The GTP shown in red is linked to the ostensible monomer of alpha tubulin tubules, and the blue GTP is connected with the ostensible monomer of beta tubulin. To strengthen the dimer framework, the medication Taxol shown in green was used. Diverse array of microtubule. The structures of microtubules differ. For example, basic cylindrical tubes or singlets MTs or cytoplasmic MTs produced by 13 protofilaments. Notwithstanding they can comprise double or triple MTs, few axonal MTs are increasingly complicated. Doubles and triplets are composed of one full MT referred to as A tubule, 1 or 2 extra tubules known as B and C tubules comprising 10 to 11 protofilaments. In both flagella and cilia, the doublets are visible, but in basal bodies and centrioles, triplets are present. Furthermore, there are populaces of microtubules, steady, long-lived microtubules, and unstable, short-lived microtubules. The function of unstable microtubules is played when cell buildings composed of MTs are rapidly assembled and disassembled. In mitosis, for instance, the cytosolic MT of cells vanishes during the interphase, followed by the tubulin forming a spindle-forming device that helps the daughter cells to separate the chromosomes equally. Spindle dismantles at the point when mitosis is finished and the interphase microtubule system changes. Rather than these fleeting, transient structures, a few cells, mostly non-replicating cells, bear stabilized systems of microtubule. The axonium and the sperm flagellum, and the small MT cluster in erythrocytes and platelets are present in these structures. Moreover, the neurons are apparently long-lived and need to create fresh connections only in adults. Neurons must keep up axons and do as such with the help of an inward core of stable microtubules. However, the disassembly of these stable structures may lead to many drastic consequences. An erythrocyte would lose its flexibility, sperm would be unable to swim, and axons would retract. Figure 4.36 Different types of microtubules. MTs form singlets, 13 protofilaments around the hollow cavity, doublets, and triplets. 113 protofilament microtubules and unfinished tubules consisting of 10 protofilaments are included in doublets and triplets. Assembly of microtubule. Tubulin subunits are amassed into an MT depending on in vitro examinations. The alpha and beta subunits spontaneously organize the dimer under controlled conditions such as perfect tubulin concenter and absence of Ca2+, which assemble in chains at high concentrations. At that point, the chain structures an assortment of an intermediate framework containing single, spiral, packed, and double ring. The rings inevitably open up to shape straight chains or protofilaments that associate one next to the other differing sheets. When a plate is broad enough, it is twisted into a tube. As a result, short tubes produced by dimers are formed. The direct incorporation of more dimers ensues continuing development. Development happens fundamentally through dimer expansion toward one side of the tubule. It is assumed that during certain microtubular capacities, for example, the anaphase spindle, the attachment of dimers at one end of the microtubule is accompanied by the loss of dimers from the opposite end. Experimental evidence: addition of tubulins at the ends of microtubules. The process of reversible polymerization of dimeric tubulin dimers takes place for the microtubule assembly, and this assembly and stability of MTs mostly depend on temperature. When a medium with a lot of tubulin dimers, GTP is boiled between 0 omicron and 37 omicron C, a polymerization reaction begins that can be evaluated with the use of the spectrophotometer Fig 4.37a. Furthermore, they are depolymerized into tubulin dimers when the MTs are cooled to a temperature of 4 degrees Celsius. In vitro, the tubulin assembles in the presence of GTP and Mg2+. GTP is very much required for the assembly of MTs. Every tubulin heterodimer binds with two GTP molecules. One GTP gets bound with alpha tubulin, and the other GTP is bound by beta tubulin. After the heterodimer has been added to MT, GTP is hydrolyzed to GTP, figure 4.37b. Basically, the process of assembly of MTs comprises three steps. First, have tubulin subunits come together to assemble protofilaments. Secondly, protofilaments are associated with forming the roof of MT, and lastly, the introduction of more subunits at bottom of protofilaments which extend the MT. The tops of the microtubes often tend to be irregular under an electron microscope because little of the elongation of protofilaments. A. B. Figure 4.37 assembling and disassembling process of microtubules MTs. Side. If, at the most, the loose tubulin quantity is greater than the critical one but smaller than that for the minus end, the installation takes place at the extreme plus end, but at the minus edge disassembly occurs. This is recognized as treadling, a phenomenon of co-current installation and disassembly. Treadling emerges when certain plus end consolidated tubulin molecule is gradually torn off the MT and ultimately loses its root on the reverse edge by depolymerization. Experimental evidence. Treadling observed in living cells by using fluorescent microtubules. The ATP and GTP hydrolysis results in a difference in the free energy of the association reactions at the plus and minus ends of the actin filament or microtubule and thus makes it possible to treadmill. The filament growth at the plus end is exactly balanced at a specific intermediate subunit concentration by the filament shrinkage at the minus end. Now, the subunits switch between the free and filamentous states quickly, while the filament's total length remains unchanged. In the form of nucleoside triphosphate hydrolysis, this steady-state treadmilling requires constant energy consumption. While the degree of treadmilling within the cell is unknown, single filament treadmilling was observed in vitro for actin, and a phenomenon that looks like treadmilling for individual microtubules can be observed in live cells by using fluorescent MTs. A cell that had been covalently connected to the fluorescent dirhodamine was injected with tubulin, rendering it fluorescent in about one tubulin subunit in 20. With a sensitive electronic camera, the fluorescence of individual microtubules was then witnessed. 1. Microtubules depolymerize, release the add tubulin at less temperature, and in the existence of GTP, repolymerize at greater temperatures. 2. Longitudinally, free tubulin dimers create brief protofilaments. Most probably, the protofilaments are volatile and rapidly linked to more stable bed plates laterally. B. A piece of 13 protofilaments includes around the MT. The MT develops through the addition of the subunits to the sides of the MT frame protofilaments. C. Free tubulin dimers on the free add tubulin monomer has GTP, red sphere, linked to a nucleotide site. Microtubule orientation. The two closures of microtubules contrast artificially. For example, they have characteristic structural polarity. Additionally, one end inherently develops or shrivels a lot quicker than the opposite end. This distinction in assembly rate can be envisioned by blending the MT-related structures found at the base of cilia, known as basal bodies, with tubulin heterodimers. Assembly of tubulin heterodimers happens at both ends, yet the MTs develop a lot quicker from one end than the other. The top of the microtubule, which is quickly growing, is regarded as the plus end and the other edge as the minus end. The lower edges of MTs are attached frequently at the centrosome, and the dynamics of MT are limited to the plus edges. The distinctive rate of growth for the plus and minus sides of MTs illustrates the diverse fundamental concentration requirements for insulation on the two sides of the MT, with a critical concentration reduced for the plus. Figure 4.38 microtubules treadmilling. Process known as the lag phase of MT installation. In this period, MT nucleation is comparatively slow. In the elongation phase, the addition of tubulin dimers is relatively fast. Finally, the growth of MTs increases to a point where free tubulin concentration becomes limiting. This results in the plateau stage, where MT mounting is proportional to the disassembly of MT. The overall critical concentration of this tubulin heterodimer is when the installation of MT is precisely equilibrated with disassembly. Once the tubulin level reaches the critical level, the TMs develop and depolymerize when the level of the tubulin drops under the critical level. So precisely, we can say that the assembly of the microtubule is composed of three stages: construction of the protofilaments by add tubulin subunits, combine protofilaments from the MT wall, and elongating the microtubule by the conjunction of more subunits to the sides of the protofilaments. However, the disassembly of MTs varies. The MT ends are accessible under shortening circumstances. The protofilaments can be depolymerized by an endless association of the tubulin subunits if separated from stabilizing. MT mounting takes place more quickly at the plus side than at the lower minus edge. The MT may bring tubulin heterodimers to the plus side and remove them from its minus side when the tubulin heterodimer level is greater than the critical concentration at the plus fork. Microtubule polymerization. The method of polymerization involves a combination of tiny molecules, recognized as monomers, chemically in order to create a big chain-like or network molecule, referred to as polymer. The monomer molecules can all be indistinguishable or constitute different compounds in 2, 3, or more. MTs have been contemplated in vivo as the polymerization operations. The fundamental phase in setting up tubular dimers in communities, known as oligomers, is to arrange them. The oligomers serve as key nuclei that can create fresh MTs, and this procedure is therefore eluded as nucleation. Once an MT is nucleated, it develops via a method referred to as elongation through the inclusion of subunits at either bottom, Fig 4.39. Initially, MT's creation is delayed relationships. GTP is also actually necessary for MT installation since the combination of GTP-bound tubulin heterodimers to promote polymerization is too fragile. In vitro studies of MT assembly using isolated centrosomes as nucleation sites revealed that some MTs could grow by polymerization while others shrink by depolymerization. As a result, some MTs effectively enlarge in comparison to the other MTs. Oligomers. TUBULIN. SUBUNITS. IN. M. ICROTUBULE. POLYM. ER. Percent. MT assembly has three stages: lag phase, elongation phase, and plateau phase. The lag phase stage is the nucleation interval period. During the elongation stage, the tubulin subunits in the solution increase quickly and decrease in their quantity. The plateau phase, during which subunits are added and removed at equal rates from the MT, will be reached if the concentration is low enough to limit assembly. Microtubule polymerization inhibitor by disrupting the MT. Drugs aimed at cell proliferation affect the MT, which causes an abnormal meiotic spindle rest in the cell cycles of the G2M phase. MT's antibiotic drugs are also renowned for their difference in mount stabilization and dynamics. At an optimum concentration, these medicinal compounds can either inhibit the polymerization of MTs, destabilize MTs and reduce the mass of the polymer MTs, or promote polymerization of MT, stabilize MTs and increase the amount of polymer. Based on the corresponding mechanism of action, the target components of MT can be divided into two groups. High destabilizing inhibitors of the MT, for example, vinca-alkaloids, vincristine, vinblastine, and colchicine. Deep mount stabilizers, taxanes, paclitaxel, and docetaxel, table 4.3. Furthermore, both MT stabilizers and destabilizers prove their effects at small levels by interacting with the MT dynamics. They are also capable of blocking meiotic development and causing apoptosis. Further, this section will be discussed in detail in the upcoming section. Classes of proved agents effects on microtubule. Taxanes, docetaxel, paclitaxel, albumin-bound paclitaxel. Stabilizing and polymerizing. Apophyllone, zixabetalone, stabilizing and polymerizing. Vinca-alkaloids, vincristine, vinblastine, vinorelbine. Destabilizing and depolymerizing. Halochondrin B depolarizing and destabilizing. Table 4.3 major classes of microtubules inhibitors. Microtubule dynamics. MTs are exceedingly vibrant tubulin fibers, which are essential for the organization of intracellular diseases such as organelle trafficking and chromosome segregation. Development and shrinking of microtubules take place both in vivo and in vitro through the growth and reduction of biochemical tubulin subunits. They develop a stabilizing framework with the development of GTP tubulin dimers at the end of MT. Underneath these are GTP-bound tubulin dimers, imparallel to GTPase tubulin action. The microtubule rapidly breaks when the stabilizing cap is dropped, in an event referred to as catastrophe, figure 4.40. A microtubule that contracts rapidly is shielded through the change from reduction to development. This event is regarded as dynamic instability and can be utilized by cells, such as to exert strength in mitosis when chromosomes are separated. Microtubules are intimately interconnected between biochemical and mechanical phenomena, a coexistence of rapid turnover and dynamic instability. This exchange process enables MTs to be looked at from a different point of view. A biochemical element considers a microtubule as a gathering of distinct subunits, which form cross-sections with two principal techniques of cooperation. The protofilaments are formed by longitudinal connections among tubulins, which are linked laterally through interactions. The tracking of MT development was empowered by single particle microscopy with a resolution of approximately 40 nanometers. Furthermore, a mechanical point of view shows MTs as an uninterrupted and strong material. The biochemical process of tubulin consists of the attachment of GTP tubulin to the MT point, the hydrolyzing of the GTP, and the discharge of phosphate, which is subsequently removed by GTP tubulin. This biochemical progress is accompanied by modifications in conformity. In fact, biochemical modifications in conformity with tubulin are mechanical. This link is discovered through all the cell and molecular biology between biochemical and mechanical developments, on the basis that conformational modifications in nucleotide hydrolysis are used to carry out mechanical work with several oligomeric proteins. The MTs are a fantastic illustration of mechanistic connection due to their structural complexity of increasing and decreasing edges. The structure of the MT end and of the tubulin dimers alter in the development loop due to the biochemical process of hydrolyzing GTP. The tubulin dimers are concentrated in the conformational process by legislative elements that regulate MT processes and combine mechanical and biochemical procedures with MT processes. The evolution of a GTP tubulin dimer into the coordination of GTP tubulin dimer in the microtubules is a perplexing procedure involving numerous conformational adjustments. Similar as the end with the stabilization cap for the growing MTs. GTP tubulin subunits enrich the wider area close to the growing end. Numerous tubulin dimers have their stabilizer cap. The widening microtubule selects several constructions from brief, sharp ends to lengthy, externally bent, and rounded constructions deciphered by cryo-electron microscopy as tubular sheets. Although most MTNs have consistent characteristics, some protophile elements are still longer than others. The distal protophile elements bend externally until beta tubulin contacts or sometimes alpha tubulin. Though most features of MTNs are consistent, some protophile elements are still longer than others. The distal protophile elements curve outward to some extent, and beta tubulin contacts alpha tubulin. The individual dimers change a minimum of 3 conformations during the MT growth and shrinkage. Such conformations may be regarded as mechanical states as a part of a cycle that stores and releases stress. In the bending or bent conformation that describes a kink of approximately 12 degrees at the interdimer border, unpolymerized GTP tubulin dimers commence. After linking to the bottom of the MT, the dimers are placed directly into an extended conformation. This method generates pressure energy in the MT grid as the tubulin dimers are kept in an unfavorable form. The dimers consolidate in the grading following GTP hydrolysis and phosphate release, leading to 3A shortening by motion of subdomain alpha tubulin, Fig 4.40. The increasing MT produces a pushing force once an impediment occurs. This power can be used to focus multiple constructions like microtubule asters and meiotic spindles. Similarly, a declining microtubule generates a holding power on items that are linked to the microtubule end. One example of this type of signature coupler is the kinetochore. Kinetochore is a significant machine to connect the chromosomes to a microtubule's increasing and declining ends. Furthermore, the spindle pushing powers emerge from the internal depletion of protophile elements that either force the coupler legitimately or predispose their spread on the microtubule cross-section. The stresses of these pushing and holding stresses can, therefore, significantly affect the dynamic instability of MTs. Hence, 4 parameters are generally measured to describe the dynamics of MT polymerization, the rates of growth polymerization shortening to polymerization, and the conversion between these two states. These parameters can be ascertained by in vitro polymerization studies. In cells, however, MT polymerization dynamics are spatiotemporally controlled by several accessory proteins along with the physical interactions with other intracellular structures. Therefore, intracellular MT polymerization dynamics are altogether increasingly unpredictable and difficult to quantify. Figure 4.40 Dynamic microtubules assembly and disassembly. 1. Change of tubulin conformational with GTP hydrolysis. In the country of GTP, green color depicts beta tubulin. In the figure, the composition of monomers alpha and beta tubulin in a tubulin dimer is straight. The GTP condition curves the boundary between tubulin dimers and causes a bending tubulin dimer. 2. MT plus ends create a band of elongated protophile elements, like a sheet, during MT insulation. At the extremes, collect GTP tubulin dimers forming GTP tubulin caps. Hydrolysis of GTP transforms GTP to GTP tubulin. GTP tubulin protophile element bends and removes the MT plus finishes during the disassembly stage in MT. The exchange of development and declining states is referred to as catastrophe and recovery. Figure 4.41 Microtubules dynamics. 1. Diagrammatic depiction of length and sidewise relationships of microtubular tubulin dimers structuring MT lattice. 2. Blood microtubule finishes 2-dimensional schematics. 3. 3 significant conformations of tubulin are illustrated by longitudinal gatherings. In a single protophile element and when unpolymerized, tubulin adopts curved conformation. The results of the reordering of alpha tubulin subunits are compacted microtubule structures. Dynamic instability. The transition between growing polymerizing and shrinking to polymerizing in vivo states of microtubules is dynamic instability. Microtubules are part of many essential cellular processes, including gametogenesis and reproduction. The fundamental importance and complexity of microtubular functions in cells lead one to study this tubulin polymer in an exciting area in which physics and biology converge. Classically, 4 parameters are used to characterize this process: the frequency of switching to a shrinking phase, catastrophe frequency, the frequency of switching to a growth phase, rescue rate, the microtubule's growth velocity during the growth phase, and the microtubule's shrinking velocity during the shrinking phase. Experimental evidence: the dynamic instability model. Both polymerization and depolarization occur concurrently. The dynamic instability model was suggested by Tim Mitchison and Mark Kirchner in 1984 to clarify that polymerization and depolarization could happen concurrently, respectively. According to this model, 2 populations of microtubules are expected, one growing in length by continuous polymerization at their plus ends and the other shrinking in length by depolymerization. The refining of both communities is that MTs are GTP linked at their plus sides, while MTs are contracted with GTP. By preventing the disposal of subunits from their plus ends, GTP tubulins protect MTs. This GTP cap offers a stable MT point for adding additional dimers. Figure 4.42. Hydrolysis of GTP tubulin causes an unstable tip. The GTP linked tubulin level is essential for the dynamic instability model. If GTP tubulin is sufficiently accessible, it shall be quickly introduced to the microtube, and a big GTP tubulin cap shall be created. When the level of GTP tubulin is reduced, the frequency of tubulin incorporation decreases. In low GTP tubulin levels near the empty point, the rate of GTP hydrolysis on the tubulin subunits exceeds the rate of addition of the new GTP tubulin, resulting in a GTP cap decrease. If the GTP cap goes off and removal of GTP binding subunits happens from the tip, MT becomes unstable. Direct proof for dynamic unsteadiness originates from the perception of individual microtubules in vitro by means of light microscopy. An individual MT can experience exchanging times of growth and shrinkage. Figure 4.42. A phenomenon known as microtubule catastrophe takes place when the MT replaces growth with shrinkage. However, in the microtubule rescue, MT either disappears completely or it can change back suddenly to the growth phase. Also, the catastrophic frequency is contrarily identified with the concentration of free tubulin. Catastrophe mostly occurs at the MT plus end at any tubulin concentration, hence the dynamic instability is more noticeable at the plus end of the MT. Dynamic instability has been exhibited in living cells utilizing video-improved differential interference differentiate microscopy and light cell fluorescence microscopy to pursue the existence cycles of individual MTs. Figure 4.42 Dynamic instability of microtubules. 1. The intracellular concentration of tubulin found in most cells 10-20 microamp favors microtubule assembly at the plus end. This end may appear frayed in slow-growing filaments due to fewer lateral interactions between the protophile elements and because GTP tubulin dimers have an inherent curve. 2. At high free GTP tubulin dimer concentrations, hydrolysis is outpaced by rapid assembly at the plus end, thereby forming a rigid GTP cap. 3. Concerted GTP hydrolysis at the plus end weakens the tubulin dimer interactions, and the protophile elements rapidly disassemble. Search and capture model. For appropriate cellular functions such as cell division, differentiation, and migration, producing a polarized microtubule organization is critically important. Microtubules themselves are highly dynamic structures, and within cells, particularly at their plus ends, this dynamic property is temporarily and spatially regulated. In order to demonstrate how microtubules create and make contact with cellular structures, a search and capture process has been proposed in which the microtubule plus ends up dynamically looking for and capturing specific sites such as meiotic kinetochores and cell cortex. In 1986, Kirchner and Mitchison introduced the exciting idea of search and capture, which is based on the dynamics of the microtubules in centrosomes and their nucleation. When microtubules develop in a discretionary manner from a centrosome, they test spaces while searching for kinetochores. Despite the fact that a solitary microtubule tests only one direction, various directions will be investigated in the end in light of the fact that various microtubules develop from a centrosome. The dynamics of the microtubules differ significantly depending on the cell region and on different stages of the cell cycle. They are both spatially and temporally regulated, and therefore their networks are polarized within cells. Kirchner and Mitchison, therefore, suggested that, because of serious perceptions of microtubule dynamics, the method of search and capture shows that microtube searches for multiple locations, chromosomes, membranes, bilayer, cell organelles, etc. communicate and then catch them while exchanged between development and shrinkage of the plus sides. Following the phase, the microtubular cytoskeleton is stabilized and reoriented. The plus ends of the microtubule show diligent growth at the leading edges of the cells when compared to microtubes in a cell with slight mobility. The kinetochore is a critical microtubule capturing cytidine mitosis. It is a structure made up of DNA and proteins, connecting microtubules to DNA in the cell cortex. Experimental evidence: the search and capture model. The search and capture model was explored in 1985 during Hill's initial theoretical research, and the medium capturing period for several microtubules was calculated to search for a certain range. Further research showed that the release period is based on the range between the kinetochore and the centrosome and utilizes only a few minutes to reach a given kinetochore for a distinctive range of 10 micrometers. The period needed for the capture of all kinetochores enhances when several kinetochores are present. The search and catch system, therefore, anticipates that it takes roughly 100 minutes to locate all kinetochores in a human body. However, kinetochores are captured by living cells within 20 minutes, which is 5 times quicker than the theory. The mild kinetic characteristics of the search and capture system are due to the fact that most microtubules develop unproposally in the cell's empty regions without DNA. If the microtubules knew where the chromosomes were, their quest would speed up. However, chromosomes make a spatial gradient, relying on RAN GT phase to show their position within the cell. RAND stabilizes microtubules after binding with GTP and encourages spindle assembly, as demonstrated in Xenophoselia egg extracts. The GTP transition to GTP is assisted on RAN by its RCC1 guanine nucleotide exchange mechanism. An enhanced focal concentration of RAN GTP is created as RCC1 binds to chromosomes. Before the GTP is hydrolyzed into GDP, RAN GTP may be removed from the chromosome and thus form a gradient of RAN GTP that reduces the chromosome distances. By expanding microtubular recovery and reducing the frequency of catastrophes, RAN GTP provides a spatial message that affects microtubule dynamics that are ranged from the chromosome. Thus, microtubules reaching a chromosomal area, where the level of RAN GTP is higher, develop and cause asymmetric microtubular allocation, with an inclination towards chromosomes. The distortion of microtubular dynamics to the kinetochore gears of the capture method. For kinetochore capture and mounting of the spindle during premature embryonic processes, this sort of partiality is crucial rather than in somatic cells, as cells at a premature point of formation are enormous and may have more positional signals than lower somatic cells for development. For early embryonic processes, this type of biasness is essential for the kinetochore capture and assembly of the spindle than for somatic cells, as cells are immense in the early formation stage and may depend more on positional symptoms for microtubular growth than smaller somatic cells. The model of search and capture is based on the centrosome of the microtubules. This model is, therefore, also regarded as a centrosomal pathway. Moreover, the local organization of microtubules in human bodies, recognized as chromosomal pathways, is influenced by genes. Interactions between chromosomes and centrosomal tubules aid the installation of the spindle. At a fully formed metaphase spindle, the kinetochores are fixed to the upper ends of microtubules, and the microtubules are usually nucleated from the lower end. Figure 4.43 The kinetochore capturing search and capture model of search and capture. B. Bias in microtubule dynamics towards the chromosomes. C. Nucleation of microtubules at the chromosomes. D. Nucleation of microtubules at the kinetochores. E. Nucleation of microtubules at spindle microtubules. F. Pivoting of microtubules around the spindle pole. G. Pivoting of kinetochore bound microtubules. H. Kinetochore movements. Drugs have been found in natural products. For example, extracted vinca alkaloids from periwinkle leaves, Catharanthus roseus. In that capacity, microtubule binding drugs are ordered into two categories: microtubule stabilizers, such as apoplones and taxanes. Another category includes colchicine and vinca alkaloids or MT stabilizers. These drugs have different locations on their target cell and are placed on microtubules depending on their binding locations. For example, in the microtubule lumen, the taxane site prevails on beta-tubulin, the vinca domain surrounding the binding site of beta-tubulin GTP. The colchicine binding location is present at the junction between alpha and beta-tubulin dimers. Microtubule stabilizing drugs. Taxanes are microtubular drugs that are placed in specific locations within the polymerized microtubule lumen. They work by binding and stabilizing GTP-bound beta-tubulin by altering their conformance to the stellar beta-tubulin-bound GTP structure. The communication among adjacent dimers results in a change in equilibrium from the soluble tubulin to the polymerized tubulin, resulting in the packaged interface microtubule phenotype. The taxanes and the taxane site binding drugs are the most consequential microtubule stabilizers, such as paclitaxel, taxol analogs, docetaxel, taxovir, and other familiar molecules. These have usually been used as cytotoxic agents that focus on many tumor kinds. Due to their ability to bind tubulin, protophylline stabilization leads to microtubule overpolymerization and eventually apoptotic death. Their cytotoxic effect is the result. Taxol, paclitaxel from the Pacific yew tree bark, Taxus brevifolia, was discovered. For the treatment of ovarian cancer, paclitaxel was approved by FDA in 1992. Tubulin assembly is encouraged by paclitaxel under all response situations, such as non-abrasive maps, less protein fixations, lower temperature, and lack of GTP causing majorly opposing tubulin polymers with shorter and exceedingly. Microtubules are depicted as green lines. Kinetochores as pink spheres on the chromosomes, gray, and centrosomes as gray spheres. RAN GTP is represented by a pink gradient in B, and augment complexes by small blue spheres in B. Dashed lines mark microtubule growth AE, microtubule pivoting F, G, and the movement of the chromosome H. In each panel, structures of interest are intensely colored, whereas the remaining parts of the spindle are pale. Regulation of microtubule dynamics, the enactment of polarized cell migration, intracellular vesicle transport of cell polarity, and chromosomal segregation during mitosis requires microtubules. Microtubules are nucleated from their organizing centers. Dynamic instability, a method of moderate plus N growth coupled with rapid depolymerization and then retrieval, is the most pervasive microtubule behavior. Although microtubule minus ends show dynamic instability, in non-organizing centers, the minus ends are mostly capped and anchored and thus do not participate much in microtubule dynamics. Hence, the maintenance of a balance between dynamically unstable and stable microtubules is controlled by various proteins that adhere either to tubulin dimers or assembled microtubules. Tubulin binding drugs.